So one of the really amazing thing that CCDI has done is, I think almost in real time, Elaine and her team are uploading data via AWS into the cloud. And one of the really early things that actually Warren had helped support as part of Kids First is that Kavatica can connect to and talk to CCDI data. And so the data actually becomes available at the same time and is interoperable. The part that's not there yet is what Javad asked, is how can I query across all that data, even though we're connected? So that's the next gap.
Okay, for the last talk before break, we're going to be moving on to discussing CCDI Federated Imaging Analysis with Ariana Familiar and Sharon Silkinson.
Thank you so much. Awesome. So I'm Shannon Silkinson. I think I'm a fresh face to some of you, and I'm really delighted to be here. I run our Federated Learning program at the National Cancer Institute, and I'm delighted to talk with Ariana Familiar, who's one of our grantees today. So we heard a lot this morning about things like Western blots and immunocytochemistry and a lot of really sort of basic science. And this is kind of the other end. This is the AI, this is the machine learning and federated learning. So I wanted to take a minute here and just kind of walk you through what that is and what do we mean when we say federated learning or that sort of thing. So there's a few real basic things, just 3 that you've got to understand about federated learning, right? So the first one is shown here. So this is a great way to share your models. You guys are super smart. You develop really cool models without sharing the data. So I've got a model. I pass it to Adam. The data stays with him. He sorts it out, and he sends me the weights back. No data, no questions asked. The other thing that's great about federated learning is it has a lot of power because, again, you can combine these multiple data sets. When I work alone at my institution, I have my catchment area, and these are my people, and this is my view. But what federated learning does is it lets us expand our horizons and look farther out and include more people sort of under our tent. And the last way, again, is this is what we talked about at the beginning, is it's a great way to train your AI models without anybody outside your organization touching and putting their fingers on your data. So again, here are some really great benefits about why that you'd want to use federated learning for your next project. Right? It reduces bias, and we talked about this because you can include more diversity in your data sets. It also really eases the privacy concerns because everybody holds on to their data. So when your patient signed a consent with you and your institution, that's still valid because I'm not taking your data. Again, we talked about the value of large data sets, and there's always a little bit of cost savings, right? Because you don't have to transfer the data. It also saves little bandwidth and cuts down on network congestion. So lots of really good reasons why everybody should use federated learning for their next project. And I wanted to give you just sort of 2 case examples. One is a project that was just released, the Cancer AI Alliance. And this is a project that comes out of both academic medical centers as well as some companies. And so what they're trying to do is use, again, these federated learning techniques to work on rare cancers or cancer clusters or things that occur in small populations. And they're doing it by creating multimodal models. So not just doing genomic data, but genomic and imaging data or genomic and clinical data. And then they're sending, again, these models across to different organizations to try to get more insight into small populations or rare diseases. The other example, I don't know if anyone's here, I'm sort of hesitant to put a UCSF example here since it's not my work, but hopefully I'll get it right, is they took advantage of federated learning to say, hey, we need a lot of diversity. There's small numbers. Let's reach across 70 different sites. Again, we'll send you our model. You train it on your data. You send it back. And when you do that, you can see on the bottom, it really improves the accuracy of the model. That is to say, this is a great example. Doesn't always work, as we might hear later, but it is really the panacea and what we're going for here. So as I leave you, I just want to all sort of stretch out. This is how we do federated learning, right? You are the smarty pants at the beginning. You hold a model. You say, hey, friends at CHOP, at the NCI, or at your institution, I'm going to send you my model. So then you send your model over here. We train it on our data. We send the model weights back to the model holder. That person aggregates the data, wash, rinse, and repeat. And at the end of the day, you have a stable model at equilibrium that's more representative of the entire population. So that's sort of what federated learning is in a nutshell. We're setting up a little network to sort of pilot this out at the NCI. Here's the first 5 stars. Some of you are represented here. If you'd like to be part of the network, we'd love to have you and add our stars to the map. But right now, I really have the honor to introduce Dr. Ariana Familiar, who's one of our grantees and who's really going to show you how federated learning works in practice. So, Ariana.
All right, so thank you so much, Shannon, for setting that up. I just actually wanted to start out by talking a little bit about our efforts over the past several years of collecting, centralizing medical imaging data that is associated with our CBTN patient database. Many different sites have put in significant effort to attempt to centralize their radiology imaging as collected as part of standard care and send it to us at CHOP as the data operating core for CBTN. So we've amassed a significantly large longitudinal database of imaging that's being used actively across different research projects. And we more recently prepared and submitted the preoperative diagnostic exams images for 370 unique CBTN subjects across different histologies, and that's released in association with the CCDI via the Cancer Data Service, and through dbGaP is released with paired clinical and genomic molecular data as well. So all of that being said, it's no mystery to pretty much everyone in this room that data sharing can be hard for various reasons. When it comes to medical imaging data in particular, there are significant technical barriers and human power barriers and bottlenecks when collecting the imaging from radiology departments at separate institutions and attempting to transfer those to a separate institution. That has largely limited the ability to generate accurate AI and ML models using imaging data, and particularly models that operate properly across different institutions, given differences in clinical protocols and in image acquisition protocols. So, as Shannon explained, federated learning is a type of infrastructure in which data is kept privately at different institutions, and instead of shipping data, the locally trained model is shipped to a central institution or an aggregating server. And in this way, sites do not have to share the data files. They are kept securely, but models, statistical and AI, ML can still nonetheless be developed. So this is a potential solution particularly for sites that have regulatory, technical, or human-based challenges for sharing and shipping data to a separate institution. Federated learning is not necessarily a new concept. However, there has been recent success in real-world contexts and real-world deployments of federated learning infrastructures. So, for example, in the pediatric brain tumor context, a recent study found that it was possible to train a deep learning model to delineate tumor regions on MRIs using federated learning, and they could also use the MRIs to classify tumor histology or the type of cancer at the given patient level. In the present project, we are just piloting here the technical feasibility of implementing federated learning. Specifically, is it feasible? What do those requirements look like? And how could we eventually use this in a real CBTN deployment context? So we're utilizing a model developed in a project led by my colleagues, Dr. Fatih Kazaruni and Dr. Kraya, in a recent study of theirs. And in this study, 201 pediatric low-grade glioma patients were selected based on their availability of clinical and diagnostic treatment-naive imaging in the CBTN database. What we're doing is we took the clinical, demographic, and treatment factors, as well as imaging extracted radiomic features. We split the data set into a training data set of 160 patients and a separate withheld testing data set of 41 patients. So that data set, importantly, is never included in model training. And we essentially built out different machine learning algorithms to predict the progression-free survival in these patients using, again, the combination of clinical and imaging-based features. We tested different statistical algorithms with different model parameterization setups and took the best performing linear and the best performing ensemble tree-based methods for further testing and implementation in a federated learning infrastructure to essentially see, can we recreate this performance in a federated learning context, and how does it vary with differences in data heterogeneity between institutions, since we know that can be a limiting factor for federated learning. So in collaboration with the NCI team, we implemented a federated infrastructure on AWS using cloud services. We utilized very lightweight virtual machines, so it was really cost effective. And we utilized an open source toolbox software toolkit called NVIDIA Flare. And so that allowed us to perform model development. Here, this is a simulation study. So there are 4 simulated sites. There's a central orchestrating aggregator, and then there's 3 separate client sites. We split or partitioned the training data between sites 1 and 2. And we then tested the developed model on site 3. So that, again, always remains withheld from training. So we first split the data 50-50, so 80 participants at a site 1 and 80 participants at site 2. We then varied the distribution of the number of samples between the 2 training sites and examined how model performance varied as a function of the different number of samples at each site, keeping the frequency of the patients who progressed to those who did not progress equivalent. Our main measure of model performance here is C-index, which ranges on a scale of 0 to 1, where 1 is perfect model predictive performance and 0.5 is random chance. So first, looking at just a 50-50 split, we essentially found that compared to the non-federated setting, the model performance declined a bit, but still compared to the reported predictive performance in the recent study. So for example, you can see here in bold the testing performance in the withheld testing data set of 41 patients. In a federated setting dropped a little bit. As the difference in the number of samples per site increased, so from left to right here, we found that the model performance declined progressively in both the training data set on the upper row and the testing data set on the bottom row. Again, this is not necessarily new. It's established that differences in number of samples for different clients in training of a federated model can cause an inability or cause difficulty in creating a model, an aggregate model that converges. However, what this does is it allows a kind of baseline for us to then take this and build out different methods of model aggregation, such as different schemes of model parameter tuning and optimization that can better account for these types of biases. And splitting the data by the type of surgery or the type of treatment that patients received also degraded model performance. So the model here essentially could not learn. Again, this is kind of a crude division of patients into separate groups. However, it underscores the need to be able to capture these types of biases in the training data set, particularly, for example, in the case of low-grade glioma, in which there can be diverse treatment options. And so, again, this just underscores both the ability of this toolkit to allow us to capture these biases, but also the need for more sophisticated model aggregation schemes. So with that, I'll just conclude by saying overall, our implementation was cost effective. It's reasonable as a solution to implement at different CBTN sites. Model development was relatively fast, even for models such as ensemble models, which can require a significant amount of model parameter tuning. That said, we still need to address issues, for example, of different acquisition protocols at different sites, which can cause variation in data quality across different sites, and be able to capture that again in this type of framework. And we are going to also include additional data types like molecular subtype information to build out a framework to do multimodal model development and, again, test different learning schemes to see if that better handles different biases and data heterogeneity across our CBTN sites. And with that, I'd just love to thank the wide cast of everyone who's been involved in this project, as well as the CBTN patients and families.
Just curious, on the NCI side, has the federated learning extended into like the MCI that we heard about recently?
And I was going to ask if either of you has examples of this outside of radiology, looking into other omics. I guess that's a little bit what you're talking about, pathology, genomics, other aspects of cancer care.
So one of the other grantees at Oregon is doing, they're studying cancer regression with tumor mutational burden. And so they're using both genomics as well as clinical data in their models.
So, Carl, the silence was a little bit deafening in the room in general when you asked about the pathology piece, and I wonder if you're so far advanced in imaging, but so few health systems have moved to digital pathology, right? Because a lot of what we're seeing is that is the holdup in pathology. Researchers have done great things, but the larger adoption beyond certain research labs and amazing work led by, Javad, the majority of health systems to aggregate enough data, they're struggling with those transformations, with less than 10% having moved to digital and clinical. So it feels like that's a huge opportunity, and it's something that we're trying to tackle, but I was wondering if that was a bit of the quiet or what your experience has been, Adam, elsewhere.
I mean, CHOP over the past year has digitized all its histology slide, but to your point, I think that's the pathology version of MRI barriers to entry to some respects. I think it's even on the CBTN side, we collect histology slides for all of our patients, but only some of the sites can actually scan them. Some of them send them to us and we scan them. I think it's, again, an opportunity that has not yet been invested in.
Yeah, and there definitely has been success in federated learning in real world using digital pathology, not necessarily in pediatric brain tumors, right, but in breast cancer and adult gliomas as well.
My name is Jeremy Schraw. I'm an assistant professor at Baylor College of Medicine, Texas Children's Hospital, and I have the privilege of moderating today's session. So I'm going to ask our four panelists to please join me at the front of the room. We can have seats in these chairs if they like. Again, ask everybody else to please take their seats. A quick note for anybody joining us remotely. I'm told that you can ask questions in the chat. I am monitoring the chat, so if you want to ask a question in there, I will do my best to make sure that it gets answered. I think we've all been bombarded with a lot of information and some really fantastic science this morning. Definitely a very quick pace, very dense information. Probably didn't have as much time with any of the presenters as you might have liked to ask questions. So a couple of folks who are joining us again for the second time today, and a couple of new faces. So the first thing that I think I'll do is I'll just ask our panelists to each give us a quick introduction.
Good afternoon. Thank you for the organizers for inviting me here today. My name is Nara Sobrera. I'm a clinical geneticist at Hopkins. I do research with rare diseases, and more specifically, a group of rare diseases that include benign and malignant tumors. So that's how I think I got here today.
Hi, I'm Suzanne McFarland. I'm an oncologist at the Children's Hospital of Philadelphia. I focus mainly on solid tumors, but also on cancer predisposition. So I do a lot of thinking about how the somatic testing we're doing can inform the germline.
I'm James Coulombe, and my training is as a developmental biologist, not a cancer researcher. And I was the NIH working group leader for the Kids First pediatric research program until I retired at the end of August.
I'm Sarah Leary. I'm from Seattle. I say my main focus is on therapeutic development for kids with brain tumors. That is still true. That used to mean early phase trials, then late phase trials, and now in my head it means collecting data as much as possible. So I think for my CBTN role, I'm working on the data operations